topic 3. Yay. The fun part of topic 3, where we get to do exercises. More exercises. Okay, so, questions, concerns, any nagging issues about yesterday's extravaganza? This is the second part of topic 3. Part 2, topic 3. Oh yeah, I think it might have gotten loaded up as 2 bits. That's how it kind of got split out last year. So yes, part 2. The continuing saga. Topic 3. Okay. Good. Well, if there are, and I meant to say this a while back, like years ago last week, but it seems like years ago. Questions, nagging issues, drop by, right? You're here. Just come by my office or come by the lab. If I'm not in the office, I'm down in the lab. My lab is in Gibb Hall, which is the building that's adjacent to where you guys have histology upstairs. So my office is downstairs, room 147 in Life Science South, and my lab is in Gibb Hall, which is the kind of kitty corner attached building, and I'm over in the corner, 125 and 126. So if I'm not in my office, I'm down there. Hopefully down there more than I am in my office. That's the way I like things. But drop by, shoot me an email. You could try calling, but you probably won't get me, so emails are probably the easiest way to, at least if you want to come by, set up an appointment, and then we'll go from there. But don't hesitate to do that. The door is always open, well, unless I'm down in the lab, but the door's open down there. So, wander about, wander in, don't get your fingers into the virus. That's all I ask. Because that's not good. So let's continue on with topic 3 and start moving into our genetic analysis and using our linkage and analysis to look for inheritance of chromosomes that might be mutated. Okay, so let's preface this. We're going to be using our VNTRs, our polymorphic regions, which we haven't talked about yet, that's next, to map chromosomes and follow them. So this is a way of following down a genetic tree the chromosome that you inherited from mom and the chromosome you inherited from dad. Okay, so we're going to be using the differences in those little polymorphic regions in those 2 chromosomes to follow the inheritance pattern of those chromosomes through the ages. Okay, so first of all, then we need to just do a little quick and dirty remembrance of our genetic inheritance patterns. You guys should have all seen this some point in time in your lives, in hopefully a genetics class, but certainly somewhere you've seen these little Punnett squares, as we call them, right? So we're going to remember there are 2 patterns, well, 3 patterns of genetic inheritance, right? So if we're talking about somatic chromosomes, remember our chromosome 1 through 22, where we have 2 copies of each. We can have 2 patterns of inheritance, either a recessive pattern, right, which means that we have to have 2 copies of a mutant gene in order to show the phenotype, right? Or dominant inheritance, where you only have to have 1 bad copy of your gene in order to show, again, the mutant phenotype, right? So the inheritance pattern obviously is the same in terms of you're still getting 1 copy from each of your parents. But if we look real quickly at these kind of examples of recessive inheritance, so now our recessive gene is the small n here, okay? And we've got two parents that both have a mutant copy of the gene, but neither of them are phenotypically showing the disease state because they only have one copy. So they've got one nice wild type copy. So they're expressing the protein of interest and we're totally fine. We call these peeps carriers, okay? So that's a word you're going to hear all the time. Somebody who has the genotype but does not show the phenotype because of it's an recessive allele. Sometimes this happens to an X-linked diseases. Okay? So when you put their chromosomes, right, remember these little Punnett square things, you can essentially assign a square to each of the chromosomes from the inheriting parent, right? And then you just do the crosses and the squares, and you see that a quarter of the time, we've got affected individuals that have inherited both mutant copies from one from mom and one from dad, right? And then you've got two that out of the four, or half of your offspring is the word I want, that are carriers, and 1/4 that are normal, right? So that's a recessive inheritance. Your dominant inheritance is going to run the same way, except now we've got our dad here who is affected, and he's got one bad copy of the allele. Mom is totally fine. Dad's only got one bad copy, but remember, in a dominant phenotype, you only need one bad copy. So again, if you do the Punnett squares, now you've got only a quarter of your babies that have a normal, completely normal phenotype. Half of them are affected. Oh, I'm sorry. You've got half that are normal and half that are affected, right? So anybody who's got the two small d's are normal, and everybody that's got inherited that bad allele from dad are showing the phenotype. So dominant inheritance is going to play a big role when we're following this linkage analysis, right? So when we get to our example a little bit later, we have to keep that in mind. In a dominant inheritance phenotype, you can have one bad allele, you can have two bad alleles. You're not going to be able to tell the difference in terms of the phenotype that that person shows, but they may have two bad copies as opposed to one bad copy. And that's going to come back to haunt us when we walk through these genetic trees. Not phylogenetics, we're not doing evolution here. Okay, so X-linked inheritance works a little bit differently, right? So remember, mom has got two copies of X, and dad only has one copy. If we have a phenotype that is associated with a mutant X chromosome, well then if dad has that mutation, obviously he is going to show that phenotype, right? So in the case of hemophilia, dad's going to show a hemophiliac phenotype. That does not preclude you from having babies, from having children. There's now easily ways to deal with and to treat hemophilia, so it's not like a devastating disease anymore like it used to be before we had clotting factors that we could give to these people. Females, right, are going to be a totally different phenomenon when we talk about X-linked diseases, okay? So males that have the X-linked mutation are immediately going to show that phenotype. Females, on the other hand, can be carriers that show absolutely no phenotype, or they can have two bad copies, in which case they would show the phenotype. But we're going to have, and we'll see this as we talk about epigenetic regulation, that we can have what we call a graded phenotype in a female that has only one bad X allele. And that's going to have to do completely with a process which we call X inactivation. So X inactivation, the evening of the dosage in females allows for inactivation of one of those X's. And it's completely random as far as which one is inactivated. And so potentially a woman can show a complete phenotype of that disease state, or it can be graded depending upon which X is inactivated and when during embryogenesis it was actually inactivated. So some of her organs may have a bad copy being expressed. Others of her organs could have a good copy being expressed. It completely depends upon at what stage during development you get that inactivation occurring. So that's kind of a cool phenomenon, which we'll talk about more later. But I just want to make you aware that women, females, can absolutely show the signs of genetic X problems as well. They're not nearly as often as males showing the phenotype, but they can do that. So again, just walking through this Punnett square issue, right? If dad has a bad X, a mutant X, then all of his daughters immediately off the bat are going to be carriers of that disease, right? Because every daughter from dad gets his X allele. Doesn't matter which of the X's that are inherited from mom in this situation, because mom is totally fine, has no mutations. However, and then obviously, right, all his sons, all their sons are going to be totally fine, because mom, again, has a clean slate in terms of X. If mom is a carrier, all of their daughters are going to be fine, although half of them, again, are also going to be carriers. And half of the sons, unfortunately, are going to inherit this mutant X from mom and will show the signs of hemophilia. The other half will get the good X and will not show signs of hemophilia. Okay, so remember, this is all about tracking. This is all about who do we get what X from, or who do we get what somatic chromosome from. And this is going to be fun to track. Okay, so how are we going to track these things, right? We can't just say, ooh, look, there's their X chromosome, and know what's going on. Or, ooh, look, there's their chromosome 22. Let's look at that, right? You're not going to be isolating. You're not going to do analysis and like punch out chromosome 22 from each of those people and sequence the whole thing, right? So you're going to have some way of tracking chromosomes. Well, this is a fabulous way of tracking chromosomes, is what we call genetic polymorphisms in the genome, okay? So right off the bat, just want to tell you that these genetic modifications, these polymorphisms, are not 99.9% of the time what causes the disease, okay? This is the way that we are tracking chromosomes. So every once in a while, the polymorphism is what causes the disease, but 99.9% of the time it's not, okay? So we have multiple different polymorphisms that we can track. And I'm just going to throw this out there too. Each one of us is different, okay? We have a different array of polymorphisms that we inherited through combinations of mom and dad, right? So this is how we are going to track individuals when we're doing forensic analysis. We're utilizing these kinds of polymorphic markers to say, that's OJ, right? That's the guy that did this. So what kinds of these things do we have? Well, we have these things called single nucleotide polymorphisms, right? So if you're looking at, this is just one strand or one chromosome here. If you're looking at individual 1, let's say in this position they might have a cytosine residue. And in this position, they have an adenosine residue. So that would be a cytidine, excuse me, an adenosine residue in the second individual. This is what's called a polymorphism, right? Some people, there's a cytidine. Some people, there's an adenosine. It happens in about 10 to 20% of any given population will have the polymorphism, right? So 80%, let's say, will have a cytidine there, whereas another 10 or 20% will have an adenosine. Doesn't matter in the least. These are generally localized in non-coding regions, so regions of the genome that aren't encoding proteins. So it's not a problem in terms of that, okay? But again, it's a way for us to recognize that individual and that individual's chromosomes. Now the cool part is you could have one chromosome where you have a C in that location and the other chromosome could be an A. So that's a way, again, that we can map and follow your chromosomes. Because one of them is going to have a cytosine there, one of them is going to have an adenine. Okay? So again, these SNPs are present in about 10 to 20% of any given population. I should point this out, right, that is much lower frequency than, for instance, a mutation that is going to cause a particular disease state, right? So BRCA1 mutation, for instance, we know it's a point mutation within the coding sequence that occurs in very small percentage of the population as opposed to one of these kind of SNPs, okay? So single nucleotide polymorphism and VNTRs, right? We kind of alluded to these before when we were talking about those signs and those repeat regions within our chromosomes. So these are little regions of repeats. They can vary in number, hence the terminology, right? So they can also vary in the size of the repeat, right? Here we've just got a dinucleotide repeat. They can be up to, say, 4 or 5 bases that are repeated in tandem. Again, remember, this is a tandem array, so it's just a big old honking section of DNA that's got this thing repeated again and again and again and again afterwards. Now, that's, again, the wonderful part about this is each person can have a variable number of these, and this is how we can track this region of the chromosome. So say, again, here, this individual has 5 copies, but this individual here can have 3 copies. Now, individual number 2 could have 3 copies on one chromosome and 10 copies on the other chromosome. Again, this is how we distinguish their 2 chromosomes. And each person can have multiple different variations on that gene. Great way to track things, right? These things are localized all over the genome. Okay, so they're not just in any chromosome, one particular place. They're all over the damn place. And we've now got a map of these polymorphic sites, right? We've done that now. We've sequenced the genome. Now we can go back and look over it and say, oh, look, there's a polymorphism there. There's a polymorphism there. There's this region of tandem repeats. We can look at this. We can use these as markers to map our chromosomes. Okay, so how would we do these? Well, we would do these by utilizing PCR, right? So if we knew the regions that were flanking these sites, whether it be a VNTR or a polymorphic region, we could PCR across that. Obviously, with the SNP, we would have to sequence it so that we know whether the polymorphism is there or not. But with the VNTRs, we can just look at how big it is, right? So we can have our flanking markers, we can look at our amplicon, and we can say, well, in this person it's 100 base pairs, in this person it's 1,000 base pairs, right? Or any variable in between that, depending upon how many repeats you got, right? So you can very easily distinguish by using PCR for these. But again, you obviously have to know flanking sequence. The other guys, these restriction fragment length polymorphisms, are a little bit more complicated in the way that you have to detect them. Okay, so restriction fragment length polymorphism, right? That says it all. This says if I take a piece of DNA and I restrict it with a given restriction endonuclease, I'm going to get a particular cutting pattern across that chromosome, depending upon how many recognition sites for that given restriction enzyme are present in that chromosome, okay? But there could be a polymorphism here, where there's just a single point mutation in that restriction site, so that the restriction enzyme now no longer recognizes that palindrome, because it's not a palindrome anymore. We've actually mutated the site. So we either have, and this can go either way, we can lose a site by mutating the sequence, or we can actually gain a site by mutating a sequence and now have a polymorphic region where most of the people wouldn't have a cut site there, but in a small percentage we now have a cut site there. So again, it can go either way of losing a site or gaining a site. Right? Obviously, when we're looking at these things, we're looking for the size of that restriction fragment. And it's going to be bigger or smaller depending upon if we're, for instance here, missing this site. That fragment length is going to be larger because there's no cut site at that position anymore. Now, how do you detect these things? Well, you detect them by doing what we call a southern blot. Okay? So we're going to cut the DNA, and we're going to run it out on one of our agarose gels, right? So we're going to use a regular old agarose gel, separate those fragments. Obviously, this fragment is going to be bigger than this. So if you restrict this person's DNA and you run it out, you're going to detect a larger fragment for their paternally inherited chromosome, and God, my brain's not functioning today, and a smaller fragment for the maternally inherited chromosome. But how do you detect that? Well, you obviously, or not so obviously, have to have a region of DNA, again, that we know the sequence, that we can design a little probe, that we can hybridize to these pieces of DNA, and that are going to hybridize and stick to that DNA and detect where that DNA is on the block. Okay, so that's what's called a southern hybridization or a southern blotting. This guy, right, Joe Southern, actually made up this technique. And I'm not lying, it actually was, I don't know if his name was Joe, but it was Dr. Southern. And there are southern blots, right? So this guy's last name was actually Southern. Swear to God. This is to detect DNA, right? So not to be undone, the guy who said, well, we can do the same thing with RNA, right? We can detect RNA on a gel just as easily. Well, he said, well, if that one's named Southern, we're going to name our RNA blots Northern, okay? And then if you want to detect proteins, well, then they're called Westerns, right? And then we don't have any Easterns, although you can't, actually, there are some kind of things where you do like Southeasterns, you probe it, and then you bind something else to it. I can't remember what you're binding when you're doing a Southeastern. When you're doing a Southwestern, you take a protein and you bind a DNA probe to it, and that's actually a Southwestern. Right, so anyway, I digress. Let's look at how this works, right? So we're chopping up our DNA, we're loading it up in the wells on a gel, and we're electrophoresing. Okay, same as we do with a regular agarose gel. Now, the key here is we take that slab gel, and we're going to, using a wicking, or you can use a current to do this, either way, you're going to transfer that DNA that's run in this gel onto a membrane. This is usually nitrocellulose. You can use, depending upon what, you can use this stuff called nitran, which is just a derivative of nitrocellulose. Essentially, what's going to happen is the DNA is going to transfer onto this surface of the nitrocellulose, and it's going to bind there because the nitrocellulose is actually charged. Okay, so the DNA is negatively charged. You're going to wick it into that positively charged nitran, and it's going to stick. Okay. Now we got our DNA on here, but what do? Well, we still don't know where our little pieces, our fragments are, right? So now we're going to take this, We're going to put it in a little, this is how we used to do it, put it in a little baggie, okay, with our probe of interest that's going to hybridize to our very specific site that we know is going to detect our bigger fragments and our smaller fragments of our polymorphic site. Now, what has to happen between here and here? Think about this for a second. We have our DNA on here. What form of DNA is this? Stranded wise. One strand, two strands. Two strands. Right, it's double stranded, right? And our probe that we're utilizing is also double stranded. So what are we going to have to do to get that to hybridize here? We're going to denature it, right? So we have to take the strands apart so that we can hybridize them back together. That's really, really essential. If you don't do that, your probe ain't going to stick, okay? So you've got to make sure you're thinking about that, right? It's just like when we're hybridizing a primer, when we're doing PCR. You got to pull the strands apart so that you can hybridize your primer so that it can extend. Same exact concept here. Now, how are we detecting this, you might ask? Well, the probe is labeled. In the old days, we used to use radioactivity to label that probe. Not so much anymore. Now we use a little enzyme called deoxygenin. You can use a fluorescent probe. Anything that will allow you to then go in and detect it using either chemiluminescence or some kind of light you shine on it. You can still use radioactivity. Not very many people do anymore. Chemiluminescence is by far a nicer way to do it, so you don't have to get rid of all your radioactive waste. So, however we want to label our probe, we're then going to now take our probe after we've washed it excessively to get off anything that's not bound very specifically, and bam, look at that. You've got hybridization to your specific genes of interest and your fragments, right? This is just a regular old Southern blot example here. So you have many, many sets of DNA that you're running. But this is just to show you what it would look like. For ours, we would have 2 bands in each because we would have mom's chromosome and dad's chromosome fragment that we were isolating here. Okay? Make sense, everybody? Okay. Now, we need one more point of interest before we start looking at our little examples here, and that is linkage. Okay, so genetic linkage analysis, as the name implies, means that whatever little polymorphic site that you're looking at is going to be inherited right alongside with your gene of interest, right? So what does that mean? It means it's linked during inheritance. That means it's really close by, okay? So in other words, we cannot have a crossover event during meiosis that could disassociate our target gene from our little polymorphic site that we want to use, okay? So crossover happens about every 100,000 base pairs. So anything that's over about 100,000 base pairs away, forget it. You're not going to use that as a linkage analysis. So, right, so if our inherited gene here, our gene of interest was our A locus, well then we could use an SNP or an RFLP or a VNTR locus that was right nearby. We're not going to have any crossing over events that are happening during meiosis. However, you can't use something way down here at the telomeric region because it's just going to have crossover events occurring in between those genes of interest and the C locus. And that's actually the way that we screen for things that are linked. Okay, so we do what's called a genetic analysis, a linkage analysis, and we say, okay, so and this is, again, I was telling you about all this Science Genetics magazine that they're doing all this linkage analysis for, God knows whatever phenotype that you want to look at. This screening is done with tens of thousands of SNPs, of single nucleotide polymorphisms, VNTRs, RFLP loci, right? So each one of these things is called a loci, okay, a locus of interest. You start out, right, if you have no idea where your gene of interest might lie, you start screening and looking at whether or not the inheritance pattern of any given polymorphism happens to track along with the disease state, right? So you're looking at a pattern of inheritance in a family. Hopefully you've got multiple generations where you've seen this disease state come up. Now we're going to track and look at genetic analysis for every single chromosome if we have no idea where this thing happens. We're going to do tens of thousands of locus analysis and see whether or not any of them match up with the pattern of inheritance of this disease. That's an enormous amount of work, right? Brute force, huge screening analysis. But you then develop essentially these, if you will, likelihoods that a given locus of interest, one of these polymorphisms, is being inherited in the same pattern and in the same chromosomal method as your disease of interest, and then you can kind of narrow them down, right? So if you say, well, this guy seems to be being inherited in the same pattern as the disease. Well, now, probably this guy is about, 100,000 base pairs away. We've got a bunch of other ones that are closer in, right? So if that one seems to be being tracked the same as the disease, well, we can move in closer and see if we can narrow it closer and closer, closer to the region that seems to really be being inherited in the same pattern. Then we start looking at what genes are encoded there, right? We've only got 30,000 of them, 21,000. It's not all that big a deal, right, to actually map out what things are in that area of interest, what genes are encoded in that area of interest. Now, you start looking at the phenotype of those genes. Well, if we know what those genes do, and something's a cytoskeletal gene, and we're looking for some liver problem, well, that's probably not the gene of interest, right? So you can map it down by the phenotype of that gene and what the function of that gene is if we know. You can map it, come up with some likely candidates of what gene of interest is actually probably mutated. Then, what do? You go back, you clone that gene into your cloning vector, you pop it into some cells, you overexpress it, you mutate it, you see whether or not it gives you that phenotype of interest that you have in your disease state. It's a very, very detailed but very distinct process, pattern that we go through to try and map this thing. This takes years, right? Let's not kid ourselves. This is like 10 to 15 years worth of information that we're talking about here, from starting from screening this thing to actually getting down to the function of a single gene that might actually be the problematic gene. This is an enormous amount of work. Many postdocs worth of many years to do this kind of thing. Yay! Let's look at an example. Okay? Now that we've hypothetically done all this, let's look at an example. First, one more point of interest that we need to think about. It really helps when we're doing these VNTR analyses or these RFLP analyses if mom and dad have what we call heterozygous inheritance, right? So if their chromosomes look different, if they have the different polymorphism, if in a VNTR, let's say one chromosome has 10 copies, another chromosome has 4 copies, so that we can track their chromosomes individually. If by bad luck, both of their chromosomes have the same exact number of repeats, let's say, and moms happen to have that same exact number as well, then we call that a wash. We can't tell the difference between the chromosomes, so we cannot use that marker to screen, okay, because we can't track anything. So at least one parent has to be heterozygous for that given marker of interest. Otherwise, it's kind of, you're dead in the water before you even start analyzing. Okay, so we think about that when we're looking at these and screening for which VNTRs or which RFLPs might be helpful for diagnosing that particular family. The other thing that we need to always keep in our heads, a given VNTR might be diagnostic for one family, but it might not be diagnostic for another family. Again, that has to do with that heterozygosity or homozygosity. If that family of interest, if two families, one has good heterozygosity in that, so we can track their chromosomes, but the other family doesn't, and they're all homozygous and they have exactly the same pattern, well then that VNTR is not going to be helpful for family B over here, but it might be helpful for family A. So not every screening technique is going to work for every single family. You kind of got to parse this through and look at it for each individual case on an individual basis. So again, pain in the butt, right?
So if the mom and dad's chromosomes can have the same number of VNTRs,
they can.
And the heterozygous chromosomes also, like by chance?
Say that one more time. I'm not exactly sure what you're asking.
When you have heterozygosity, does that always mean that the VNTR regions are going to be different lengths? Because couldn't they like coincidentally be the same length as well?
They could absolutely be the same length, but we need to be able to tell the difference between their chromosomes. Yep, and at least for one parent, and we'll see some examples here in a second where they actually don't have differences, but we can still track it because we've got heterozygosity for one.
Oh, okay,
that's where it comes into play. Yep, you got to have at least one that's got heterozygosity. Okay, so let's look at this example, and we're going to walk through this together, and then we're going to have a exercise. Fabulous exercise. Okay, so this is easy breezy. We have an X-linked disease here, okay? And we're doing RFLP analysis, right? So we're doing a little southern blot. We're going to restrict the DNA from mom and dad, from the kids. We're going to run them out on gels, and we're going to probe, using our probe of interest, to see how big those areas of the chromosome are, right? So remember, and this is what we're using here. We're using our Hindi3 enzyme. We're cutting the chromosome of interest. And the probe region is going to be here. Okay, so if we have the polymorphic site, it's going to cut, right? So here we're actually introducing a cut site. And if we don't happen to have that polymorphic region, it's not going to cut. And so we can tell whether a chromosome has a polymorphism, because it's going to have a smaller fragment size, or whether it doesn't have a polymorphism. So remember, again, polymorphism doesn't mean disease, right? It just is a way to track the gene of interest, okay? Right, so we're X-linked. Mama has 2 copies, Dad has 1 copy, right? So here's a little bit of genetic tree lingo, okay? Squares are dads, circles are moms, okay? If you got a dot in the middle of your piece, that means you're a carrier. And if you're filled in, that means you have the phenotypic disease state, right? So you can tell whether or not that person has the disease state, okay? So here we have, and right, remember, we're running our DNA. So this is the top of the gel, this is the bottom of the gel. We're running in this direction, so the larger fragment's going to come up on the top and the smaller fragment on the bottom, right? So that guy has a disease state, right? That's a guy, so that means he has 1 X, right? So mom has 2 X's, one of them is mutant. Which one? Her 3KB is a bad copy, exactly, right? So what does that mean about this one? should be okay, right? So, it is okay, and we know that because son number 2 over here, who does not show the disease state, has inherited that X from mom, okay? And he's totally fine. So that X is totally fine. So what about this babe over here, okay? Where's this come from? And this comes from dad, right? So what does that mean about dad's 3 kb fragment? It's okay, right. So what does that mean? 3 kb does not equal bad, right? 3 kb just means 3 kb, just means you got the polymorphism. Doesn't mean it's bad, right? It has to be linked to an individual, right? Okay, so mom's 3 kb is bad. Dad's 3 kb is okay. This Babala over here has mom's 4 kb, okay. Dad's 3 kb, okay. What about this one? Why is there only one band?
She has 2 copies of 3
kb. Right. So what does that mean about her?
She's a carrier.
Oh, tricked you. Almost. Didn't put a dot in the dot. Right. Yeah, so she is a carrier. Exactly. Oh, we didn't know yet. Right. Why would we have known? She's phenotypically totally fine. So, I tricked you. But I didn't. You didn't. You fall for it at all. Okay. Good. That's all there is to it, right? We're just tracking, again, where did we get what chromosome from? Follow it along. We follow it in, and normally, and this is a really good way to think about this, work from inside out, okay? Work from the middle, where the last generation is going to be sitting, and work outwards to find which chromosome have they inherited from which parent, okay? So, now we're going to get a little bit tricky here. Ooh, you thought it was all going to be easy. But it's not, right? So, we're going to set this up, and then we're going to work in teams to figure out the questions down here, okay? So, we've got 4 different BNTR phenotypes now, okay? This is just copies of repeats, okay? So, allele number 1, right? If you have this allele, you have 6 copies of the repeat. Allele 2, you have 4 copies. Allele 3, you have 3. And allele 4, you have 1. Okay? I'm going to hire and actually PCR across 1 copy. It must be a fairly large. Anyway, that's neither here nor there. You have 3 generations. Okay? You got grandmas and grandpas out at the edges. You got mom and dad in the middle. And you have Gen X, Gen 3 in the middle here. Okay, once again, we're doing PCR now across this VNTR, and we're running the fragments from top to bottom. So the largest allele number 1 that has the largest number of copies is going to be the biggest fragment, so that's going to be on the top of the jump. Okay, so you track down under each individual, right? So Grandpa A over here, this is his pattern. Grandma B, this is her pattern, and so on. The dashed lines are just trying to help you track in from each generation, okay? So, you got 4 things you need to do. First of all, and don't shout it out, and work in your groups. Question number 1, is this a dominant or recessive trait? Question number 2, for each parent, that's the generation 2 guys here, I want you to tell me, if any, which of their alleles seem to be linked to a mutant gene. Okay, so remember, don't get tripped up. It could be different numbers for different parents, right? Now, I also want you to look at, while you're doing that, and you kind of have to, look at whether or not in generation 1, you can figure out which are the bad copies for generation 1, 2. you should be able to do that. There are a couple, I'll give you a hint, there are a couple that you're not probably going to be able to figure out whether they're mutants or not. And then the last thing we need to look at is our little baby boy here, baby B, and we're trying to figure out, right, this is a late onset disease state. And we want to figure out whether or not baby boy B, say that 5 times fast, is potentially going to develop this disease, given the parameter of this family inheritance and what genes he has inherited, right? Has he, in other words, inherited bad copies of the disease? Renee?
So C, D, and E, are they all, we know that they cannot have.
We know that they're older, right? So this is a late baby. Let's just call it a late baby. He's got very, very much older siblings that have already either manifested the disease or not. Good question. Okay? And then last thing, right, this should be an easy thing to answer. Does this BNTR, will it be helpful for all families with a history of heart disease, which is what we're looking at? Okay? So, let's say, we have 2 options. We can take a break now and then come back and do this, or we can do it and then take a break. What do you want to do?
Do it.
Do it! Yes! Good job. That's exactly what I wanted to hear. All right, do this. Let's do this.
Affected parents and not all of their children have the disease.
Right. Every gen has some people that have this phenotype. Has a real good indicator that it's a dominant phenotype. You've only got to have one bad gene.
A recessive phenotype with both parents having it, don't have to have it.
Well, if you have a recessive phenotype where both parents have it, then all of your genes are bad. So yeah. But that's not going to be highly likely, and then everybody would have it, right? So because that's not the case, you're going to dominate both. Okay. So for each parent, which alleles seem to be linked to the mutant gene? Well, let's start in the middle. Because the best part's always in the middle. Think about cookies. Think about pie. C, D, and E, right? We can tell flat off the bat if we know this is dominant, right? We're assuming it's dominant. C, D, and E children are all totally fine. They have the same exact genotype, okay? So, in terms of this BNTR. So, what can we tell about these 2 chromosomes? Where did they inherit this from? Mom. Right. So, what does that mean about this? It's okay, right? Not mutant. So what does that mean about this? So that's our bad allele from mom, is allele number 3, right? Now this, allele number 2, they inherited from dad. It seems to be okay, yeah? So that means this allele is bad for dad. Bad for dad. Okay, so if this allele, so let's track it back now. Let's go farther out. If this allele is okay for dad, well, that means he inherited this allele, what is that, 2, from his mom. So what does that mean about this? Bad, right. Now, he inherited this guy from his dad. What does that mean about this?
Could be either.
Could be either, exactly, right? We don't know. This is one that we're not sure about. Okay, let's do the other way, right? So this allele we got from mom, that's okay. So that means she inherited this from dad. So that means this allele is okay from dad. That means this allele is mutant from dad. She got allele 3 from her mom. So that means this is mutant. So what do we know about this? Bupkis. Yeah. Don't know, don't care. We know which one was bad. That's all we need to know. Okay? So, let's recap. Allele 3, bad from mom. Allele 4, bad from dad. Okay? So, let's walk through the rest of these chillins. Starting with G. alleles are bad here? Both. Yeah. F and G both have 2 bad alleles, right? A, what do we know? 3 is bad. 2's okay, right? Because that's the one that these guys inherited from dad, and it's okay. So what about baby boy B? Poor little thing. Likelihood is he's going to get heart disease too. Hopefully we'll be able to treat him and he'll be okay. Now, is this BNTR going to work for everybody? Yeah, probably not, right? Likelihood that somebody's got the same exact disease inheritance pattern as this set of peeps? Probably not. Yay, fabulous. Good job, everybody. Let's pat yourselves. Lean back, patting everybody. Do it. Can you do that? That's something you should do. Wilson.
So for the future for F and G, if they were to have kids, would they automatically transfer the disease because they're homozygous for both?
They would automatically transfer the disease, yep.
So their kids wouldn't, they would just know.
SOL.
If we were presented with all this information straight from the get-go, we wouldn't necessarily need to answer question 2 to answer question 3, right? Because B has the same genotype as A, and we already know that A expressed the disease phenotype, right?
Fair enough, except you better look back and make sure
Right, we
that
still
they're
wanna-
coming from the same parent. Right, you better, you should do it. You should do it anyway.
But just because in this rare
Is there
instance,
a risk that,
in
you're
this rare
absolutely
instance,
right, in this instance where
as parent
we're
A and
doing
parent
a
B.
diagnostic and we already know someone who has that same exact genotype has a mutation and that you've got this phenotypic inheritance pattern, yes, you're
Well, worse
absolutely
is-
right.
parent A and parent B, they all have unique alleles.
Correct.
And none of them have the same
Correct.
BNTR numbers. That's why.
That's why you want to make sure and track it back and make sure that we're getting that same.
If their parents did have one of the same alleles, would you still be able to do this?
Probably yes, because you could probably track it in terms of they got one of the 3s from one parent versus the other parent. If that one had, not necessarily in this case,
Okay.
but depending upon the whole setup and the whole inheritance pattern, you might be able to, you might not be. And again, that's going to be completely dependent upon what phenotypic traits you've got and how much disparity you have between the 2 parents and their grandparents, right? The farther back, and this makes a lot of sense, right? The farther back in the tree we can go, the more information we have, the more likelihood that a given BNTR is going to be helpful. Sure. Yay! Let's take a 10-minute break. We're back here at 27 after.
How do BNTRs mutate the course of the person's life?